Hello, thanks for joining me online, and I want to spend about 30 minutes going over the material for the final exam so that you'll, and this is, as you look at these individual questions or comments about, know these differences, recognize differences, or the specific questions, very much like midterm 3 and midterm 4, this will be the material that I'll be using to develop the final exam. So if you can answer these questions, then you'll do quite well on the final. So I'm going to go all the way back to the beginning of the semester, except for handout 1. That material gets covered later in the semester. And so the first section is going to be on handouts 2 through 5. Now, I'm taking, these questions actually came directly from the midterms that we've had up to this point, midterm 1 through 3 and then 4, which you'll be taking Monday. So if you look back at the midterms, you're going to recognize these questions. And I'm not asking all the material that was on each of the midterms. There simply isn't enough time or space in a 100-point final exam. So you'll recognize this information if you go back to your individual midterms, see how they were graded, and you're going to do pretty well on this. So from handouts 2 through 5, you'll notice that there are let's see now, handout 2 through 5. Okay, and I might have this kind of name funny, but you're going to recognize this material. So as we indicated for carbohydrates, I wanted you to know the differences between ketones and aldehydes. And so we talked about dihydroxyacetone phosphate and glyceraldehyde 3-phosphate being the smallest triose and aldehyde respectively. And so be able to recognize those and know the differences between ketones and aldehydes. And early on, I really stressed the importance of structures, and I'll continue that through the final exam. So, once again, I want you to be able to recognize structures for lactate, pyruvate, oxaloacetate, and alpha-ketoglutarate. So we have our 3 carbons, 4 carbon, and then 5 carbon. And as you know, I'm not asking you to draw the structures, but I certainly think you should be able to recognize those. And so just for carbohydrate metabolism, those are the key points I want you guys to know. And I think they're some of the most important, just talking about structure. And jumping to lipids, I want you to go ahead and look back at the notes and be able to list the common names for the fatty acids. And also, for example, if I list stearic acid, then I want you to know, be able to recognize that it's 18 carbons, no double bonds, or if it's oleic acid, as an example, it'd be 18 carbons, 1 double bond, and then use the delta system to tell me where that double bond is. And so as we increase in double bonds, if it was linoleic acid, you would tell me that it was delta 9, 12, and so on. So we're going to really look for the common fatty acids. I want you to remember and be able to tell me how carbon number and double bonds affect melting points of lipids. This is important for the functionality of the tissue as well as for the organoleptic properties of the meat. Now, more structures. I recognize structures for alanine, aspartate, glutamate, and glutamine. And again, it's 3 carbon, 4 carbon, and then 2 5-carbons that are derived from alpha-ketoglutarate. So you should be able to recognize this. And the one difference here is that glutamine has 2 amino groups. So it should, if you see this, it should really jump out at you. But at this point, you should be able to recognize that. And I'm just going to ask some, we've covered a lot of material on digestion of carbohydrates and fats and lipids, and proteins. And I'm going to be just asking general questions, and particularly I want you to be able to compare differences between monogastrics such as pigs and poultry and in ruminants, sheep and cattle, goats, because there's some real differences because of the rumen in how all of these materials are digested and absorbed. So I think I got the numbering off here. That doesn't surprise me. But we talked about pathways involved in carbohydrate metabolism, lipid metabolism, protein. And so for carbohydrate metabolism, what are the enzymes that regulate glycolysis? And I focused, of course, on hexokinase and 6-phosphofructokinase, and how these are regulated. And of course, one of my favorite means of regulating 6-phosphofructokinase is citrate, which of course means that you've got to focus on those tissues that have mitochondria, red muscle, cardiac muscle, liver. That's where citrate is only important. So put an exclamation point behind mitochondria. And one of the things I want you to remember or recall is how if you, when you're going from glucose to glucose 6-phosphate, and that's the hexokinase reaction, that glucose 6-phosphate strongly feeds back and inhibits hexokinase. And then because this is in equilibrium with fructose 6-phosphate, and then there's the PFK reaction going to fructose 1,6 bisphosphate. If citrate and ATP, as they feed back and inhibit 6-PFK, then that forces fructose 6-phosphate back to glucose 6-phosphate, and you get a kind of a double inhibition just because you've inhibited the 6-PFK. Then you're going to ultimately wind up inhibiting a hexokinase, and the glucose is going to not be taken up and remain in the blood for use by other tissues. And I come back to this point, I made it a couple times in our lectures and exams, that we need to be able to regenerate NAD in order for glycolysis to occur. And it's jumping again to ruminants. Why is propionate production so important for ruminants? And where does propionate come from? Of course, this is critical for the production of glucose via gluconeogenesis. Well, my writing's no better now than it has been all semester. Gluconeogenesis. So ruminants are really dependent on propionate production. And we spent quite a bit of time talking about glycogen synthesis and the degradation. And I'm looking just for an overall pathway here. And I drew what was essentially I don't really want to, cycles maybe not the right term, but you can draw this kind of in a circular fashion where you start with glucose 1-phosphate and make glycogen, and then when you degrade glycogen, you have glucose 1-phosphate again. So, but be able to talk about that. I really want you to recall how epinephrine, left out a word here, affects or controls glycogen synthesis and degradation. That epinephrine is going to basically inhibit synthesis but promote degradation. And so if you understand the importance of cyclic AMP and protein kinase A, then you're doing really well here. So, and again, because there's so much material, there's only 100 points, I'll be asking general questions about this, the overall regulation of synthesis and degradation of glycogen. And so this would be carbohydrate metabolism. Then handout 7 and 8, I believe this is just handout 6 for glucose metabolism, glycogen. So we jump to lipid metabolism, my favorite topic. But what is the primary substrate? Let me take this off, it's clunking. What is this primary substrate for fatty acid biosynthesis in ruminants? Of course, it's acetate. It's one of the VFA. Where does this come from? Okay. Now, all of you did well with this part, and so you'll basically be going back and review this material, but the flow charts you gave me were great. Be able to describe the overall pathway of conversion of glucose, lactate, and acetate to cytosolic acetyl-CoA. And of course, the importance of this is your substrate for acetyl-CoA carboxylase. Which feeds in, it produces malonyl-CoA, and this provides substrate for fatty acid synthase. So the enzyme complex. Enzyme, okay. And it's really important that you understand that NADPH is required for the fatty acid synthase complex. So again, be able to describe the three pathways that are responsible for the synthesis of NADPH. Now, the problem with me recording this is you can't raise your hand and ask questions about it. But I think if you go back to your notes, go back to the midterms, then you're going to see all of this information provided for you. So studying the midterm is hint, 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 hint. It's a great way to study for the final exam. The questions won't be exactly the same questions as on the final, as on the midterms, but they're going to be pretty close. So, okay. And this next one, I mean, if you didn't know this was coming, then I'm really shocked because we've talked so much about this. Be able to outline the conversion of stearic acid to docosahexaenoic acid, or DHA, and know the desaturases. And you don't have to name the specific elongases, but well, list what do I want to say? The elongation steps and in their appropriate place. So, if you've got to go to something that has 18 carbons, it finally winds up with 22 carbons. And I realized specifically be able to differentiate between algae and animals, just as you did on the midterm. For this, the next one, be able to describe the overall pathway for triacylglycerol biosynthesis. And several of you, I said, oh, I need more detail. So you lost a point or two because of that. So be sure to list that, we go SN1, then SN2, and so forth until we get to triacylglycerol. So make sure you give me enough detail as you address that question. And again, I don't think you should have any problem with it. Maybe those that didn't list all the steps didn't realize that I wanted more information, but this time you know, so I think you'll be okay. So just a couple of questions on lipolysis, and again, similar to what I asked you on the midterm. Where does lipolysis occur? In what cellular compartment? And you should be able to tell me that this is in the cytoplasm. And then, of course, what are the products of lipolysis? And in this one, really the primary products, I took a lot of different answers, but to be correct, really specific, we're really looking at the fact that the hormone-sensitive lipase especially produces the 2 monoacylglycerols and 2 non-esterified fatty acids, or just fatty acids. That hormone-sensitive lipase doesn't really go all the way to the free fatty acids and glycerol. And so for the final, I want you to know that, okay, we only really get a partial hydrolysis of triacylglycerols so we wind up with a 2 monoacylglycerol. Now, for fatty acid oxidation, we've talked about this a couple of times. Okay, so where does beta-oxidation occur? And again, this is mitochondria. So, by extension, only those tissues that have mitochondria can actually oxidize fatty acids. So if the cell doesn't have mitochondria, then it can't get energy from fatty acids. And what are the primary products of beta-oxidation itself? Well, primarily, you've got fatty acids going to go to a fatty acyl-CoA, and then depending on how many carbons, it's going to produce a large number of acetyl-CoAs. And by now, so many, we've talked about this so often, you realize that this is going to go to citrate and ultimately to CO2, but really we're talking about acetyl-CoA production. So, and then later I'll talk about, okay, we use, if it's the liver, it's going to make more acetyl-CoA than it can oxidize, so this ultimately is going to go to ketone bodies, but that's later. Now, much more recently, we've talked about the general process of fatty acid peroxidation, and on your handout, in one pathway I showed the structures, another was just a flow chart, and I'm really just interested in the flow chart. If you understand that, I think that's great. Ketone body synthesis. Again, more recent information. Where does ketone body synthesis occur in the cell? Which tissue? And if I didn't make it really clear in the material, this is going to occur only in the liver. That other cell types, other tissues, do convert fatty acids to acetyl-CoA if they have mitochondria, but they don't produce them in such high amounts that ketone bodies will be produced, and they don't really have the enzyme systems for this. So we're really looking at the liver. Which ketone bodies which tissues use ketone bodies for energy? Well, if they have mitochondria, just ask yourself, do they have mitochondria? Then those tissues. And so red muscle, which would be type 1, and we didn't really say specifically, but there are also the type 2A, because it has some mitochondria. And then, of course, cardiac muscle. And why are ketone bodies preferred over fatty acids? And we've really just gone over that. Of course, they're water soluble, and they're activated in the mitochondria. So keep coming back to the mitochondria, how important they are for metabolism. And then we jump to amino acids again. Because we're talking about ketone bodies, we distinguish between ketogenic and glucogenic amino acids. And which amino acids are strictly ketogenic? Well, it's the two L's. Leucine and lysine. So no part of their skeleton, carbon skeleton, can go to derive a TCA cycle intermediate, a glycolytic intermediate. That's all that they can produce. And then, consistent with that, then be able to describe an amino acid can be both ketogenic and glucogenic. And as I've tried to stress, it's when I say if they're both, then a portion of the amino acid can go to a ketone, and then a portion can go to a TCA, into the TCA cycle, or glycolytic intermediate. Okay, and just a little bit on phospholipids. And we've talked about, there are branch points in triacylglycerol biosynthesis where phospholipid synthesis occurs. So as you go from let's see, how do I want to put this? Well, lysophosphatidic acid to phosphatidic acid to diacylglycerol, and then ultimately to a triacylglycerol. We've talked about the fact that a diacylglycerol can go to phospholipids and phosphatidic acid can go to phospholipids. Okay, so the overall pathway, when you start with a glycerol 3-phosphate and add in a fatty acyl-CoA, and then the CoA comes off, then you get lysophosphatidic acid, you add in another fatty acyl-CoA, again you lose the CoA group, phosphatidic acid, and then the final step here to get to triacylglycerols is adding in one more fatty acyl-CoA, losing another coenzyme A group to get the triacylglycerol. But again, phospholipid synthesis can just use either phosphatidic acid to produce one class of phospholipids, or it can use diacylglycerol to produce the other class of phospholipids. And this would include the most important one. When the diacylglycerol goes to phospholipid, one of the main ones is phosphatidylcholine, or its general name is lecithin. So I'm not asking enzyme names, but if you know the individual steps, I've just covered triacylglycerol biosynthesis and phospholipid synthesis in a fairly easy flow chart, and so that's something I want you to be able to reproduce as well. And in a second question associated with phospholipids is just, what is the general importance of phospholipids? How does this differ from triacylglycerol? Well, triacylglycerol are for energy storage, primarily. You could also list insulation, but this is really an important point. And phospholipids, if you remember, these are going to wind up primarily in membranes. Okay. And then we jump to, in the last set of handouts, we jump to nitrogen metabolism, and start with what happens when we have excess amino acid nitrogen. How's it excreted? Well, it depends on species. So we talked about urea, uric acid, ammonia. That's supposed to be ammonia. And which species are going to produce urea, which uric acid, which ammonia, and which basically is all of them. And spend a little bit of time on the transamination reaction. This is so important for the production of the non-essential amino acids, and which is involved in producing the non-essential amino acids, moving excess nitrogen from one tissue to another, such as from muscle to the liver. And so just be able to describe this overall pathway for amino acid transaminases, and really understanding why alpha-ketoglutarate and glutamate are important in transamination. And we'll put a period there, because these two are involved one way or another in all transaminases, whether it's for transaminating alanine or transaminating the branched chain amino acids. You're going to be, if you're producing alanine, you're going to use glutamate. If you're removing nitrogen from alanine to produce pyruvate, you're going to transfer that nitrogen to alpha-ketoglutarate. So those two are involved in this equilibrium reaction. So this is really important about this inter-organ amino acid exchange. And some very specific questions about that. Which amino acids are not metabolized by the liver? Well, you guys should remember it's the branched chain amino acids, and the liver is lacking in the branched chain amino acid transaminase. And this is going to conserve the branched chain amino acids so that they can go to other tissues for protein synthesis. If the liver used them all up, then muscle and other tissues would be out of luck. You couldn't produce muscle proteins. So, and then what does the liver do with the excess amino acid nitrogen? And of course, it's going to use that to produce urea. Okay, so that's the fed state. The post-absorptive state or fasted state is pretty similar. Which two amino acids account for most of the amino nitrogen released by muscle? Well, that's pretty easy. Alanine and glutamine. And these are going to go primarily to, they can go to liver, to the small intestine. And so which amino acids can muscle transaminate that the liver cannot? Well, the branched chain amino acids. So, but only the excess. They're not going to use branched chain amino acids. They're not going to transaminase. Let me say that differently. The muscle will not transaminate the branched chain amino acids to their alpha keto acids if they need those amino acids for protein synthesis. Okay. And I want to kind of wrap things up. We talked about urea synthesis, and I tried to put it in context of the fact that it's a way to excrete nitrogen, but in ruminants it's an important source of nitrogen for protein synthesis. So first of all, where does urea synthesis occur? Well, it's in the liver and primarily, and we talked about the fact that the kidneys could be involved, but we're really looking primarily at activity in liver. And again, just want you to be able to describe the overall pathway of urea synthesis. So cellular compartments, the mitochondria is essential here for the first steps, and then be able to list the intermediates like carbamyl phosphate, which is going to be produced in the mitochondria, and all the way down to arginine, and then to urea. So just be able to describe the overall process there, and also be able to include, urea has two amino groups, and list the sources, the two sources for those amino groups. One's going to come with carbamyl phosphate, and then the aspartate's going to feed in the other amino group. And then, putting this in a more practical sense for animal production, talk about rumen microbes as protein sources for the ruminants. What is microbial crude protein? We talked about that quite a bit. What's the contribution of microbial crude protein to the diet? And of course, we're talking about ruminants here. This has very little impact in non-ruminants. And bringing urea back into the picture, what is the relationship between urea and microbial crude protein? And go back and look at some of the figures we had. What are the possible sources of urea in the rumen? And remember things like saliva and just absorption. So there are other sources of urea, and one of those is just in the feed, that will actually add urea to the feed in cattle, and that nitrogen can be used for the microbial crude protein synthesis. And just in general, be able to describe how ammonia is incorporated into microbial crude protein. You need a carbon source and the nitrogen, and be able to draw a flow chart for that, or just a simple description of how the microbial crude protein actually gets synthesized from the ammonia nitrogen, which ultimately can come from urea. And then finally, today's lecture, I'll be going over these details. So, talk about beta agonists and muscle growth. Try to bring together everything in today's lecture. So, which hormones do the beta agonists resemble? Well, epinephrine, norepinephrine. Which muscle fiber types are most consistently affected by these beta agonists? I'll talk about these, especially the type 2B, the large glycolytic muscle fibers. What's the effect of beta agonists on marbling and tenderness? And I'm going to spend some time talking about beta agonists, how they actually increase muscle growth. So I'll be talking about synthesis of proteins and degradation, and how the beta agonists can actually impact both of those and give you a pretty profound muscle growth because of that. And then finally, because marbling is my favorite thing in the whole world, I'll talk about marbling scores and beef quality. And this is actually going back and reviewing material that was in handout 1. What's the relationship between intramuscular lipid and flavor, desirability of beef? and really talk about just spend a few minutes on what exactly is marbling. And so we'll get to wrap up with that information. And so we'll cover the details in today's lecture. You know, what is marbling? And basically it's just a very specialized fat depot. And so we'll be able to summarize that today and kind of have a little fun with today's lecture. And that's basically how the review material, you know, if you can go through and follow what I've written down and go back to the previous midterms, I think you're going to have very little trouble with the final exam. So I'll be seeing you guys later. Thanks. Bye.